Nowadays, AI is a huge umbrella for what it is. I like to say that we use whatever tools are necessary to get the job done. And that spans math, physics, biology, data science, AI. And what we find is that AI is absolutely great at doing some types of analyses that are very standard and straightforward. And it essentially lets us explore the space of possibilities much faster.
Welcome to Innovate Law. I am Antoinette Konski, an intellectual property partner with Foley and Lardner. Join me as we learn from the minds who are shaping the ever-evolving world of the innovation landscape. And today's guest is Dr. Tal Einav. Dr. Einav is a leading figure in virology and infectious diseases, recognized for his groundbreaking research on viral pathogenesis and host virus interactions. Holding esteemed positions at institutes like the La Jolla Institute for Immunology and the University of California, San Diego, Dr. Einav's work spans diverse viral infections from hepatitis C to emerging threats like Zika and SARS-CoV-2. His interdisciplinary approach combines molecular virology, immunology, and computational biology, driving advances in antiviral therapies and vaccine strategies. He is a visionary leader and mentor. His contributions shape infectious disease research and inspire the next generation of scientists. So Tal, you're a physicist by training. How did you get into viral research and immunology?
The fun question. It's the question of how do we all get to where we are going today? And the answer was a series of very lucky and fun hops. So I was trained as a physicist, and in grad school, when I was choosing my lab at Caltech, I talked to everybody on campus that I could and just asked them, what would research be like in your lab? And the one that got me, the one where I resonated the most, was my future advisor, Rob Phillips, who told me that physics has the best tools, but biology has the best problems. So why not come and play over in his side of the field? And Rob was just like me. He was a biophysicist, so a physicist turned biologist. And that was really my introduction into biology, where I started from the ground up, asked all the fun, embarrassing questions, learned my way up. And then at the end of my PhD, I did another project with Pamela Bjorkman, also at Caltech, on HIV antibodies. And I just felt like this project resonated with all of my skill sets, the math, the physics, the computer science, the biology. It pushed them all forward in a way that was very me. And so I said, this is going to be my future. So in my postdoc, I hopped into immunology, which is where I am today. Although every one of those steps is still in my DNA, if you will, and builds upon how I think about problems every day.
That's really interesting. So you went from being a physicist to a biologist, and you mentioned HIV research. But what's kind of guiding your research, and what are the big problems you're looking at today?
So as a physicist, I would say that the biggest asset I bring to the field is that I ask different questions from what biologists normally look at. And one of the things that we like to do, especially in physics, is to make models to understand how systems work. And so in my PhD, that was understanding how bacteria work. Now in my current job, it's understanding how people work. And people, as it turns out, are very complex. There's all these nuances. Everyone acts differently. If we think about vaccination, which is what my lab does, then for flu vaccination, for example, about 1/3 of people each year who get the flu vaccine have a very weak response to it. And we don't know why. The other 2/3 respond fantastically, but there's this 1/3 that is different every year. So if you're in this 1/3 one year, you might not be in the 1/3 the next year or the year after that. But for some reason, there's this pretty sizable part of the population that just doesn't respond as strongly as others. And so in a biological setting, you might ask, well, that's interesting. What can we do? Does it happen year after year? What are the features that actually correspond to these kind of responses that make them weaker or stronger? In my lab, we try and predict what happens ahead of time. So we try to take people at the beginning of each flu season, and we try to understand who are going to be these 30%. How do we think they'll respond? What do we think would have happened if they could have potentially taken a different vaccine rather than the vaccine that they were given? And could that help them mount stronger responses? So it's a different way of thinking, really.
So your research, just to state it another way, is really, in one aspect, personalized medicine. If you're looking...
Absolutely.
Yeah. Well, that's really great. I know that's just a emerging field of science and technology that really is... We're seeing a lot of advances lately. So with respect to vaccine development and understanding how each individual may respond to that, how does your approach differ from what others are doing now and have done in the past?
Okay, great question. So there are two major differences. The first one is that biologists in general love heterogeneity. They embrace the differences and the uniqueness that makes each of us our own special self. And they like to point out, like, look at the various responses that we get. Some are weak, some are strong. Some people do great. Some people do great with other vaccine types. We try to make unifying principles. So what is one model that can describe how everybody acts? And maybe you need some parameters in there to describe what is a person. Is it their age? Is it their sex? Is it how their antibody levels look like before vaccination? But what are the key parameters that we need to describe a person? And I think about everything like a physicist does. So in physics, we have, for example, the ideal gas law, which tells us that if you know the pressure, temperature, and volume of an ideal gas, you know everything about it. It doesn't matter what its shape is. It doesn't matter what its composition is. The point is that once you know these three key things, you know how the gas will behave. And so we're looking for the analogous key variables for humans in terms of how they respond to vaccines. So that's one big difference. The other one is that we are swimming in data nowadays. I mean, biologists have gotten so good at creating treasure troves of data sets that it's gotten to the point where it's hard to sift through all of it by yourself. You can look at this gigantic Excel file with 100,000 measurements, but you can't keep them all in your head at the same time. So we're now at this point where we need to be able to have methods that can sift through these data sets and slice them up in very creative ways, not just in the ways that we've used to done, but looking at different unique slices that reveal patterns that used to be hidden beforehand. So what we do is we ask different questions. We ask, what is a person? How do they respond? What are their key features? And then we use that in all of the myriad data sets that we have to understand how robust are these relationships? What really do we need? What strong statements can we make about each person given the data that we have about them?
That's fascinating. So also, if I'm hearing you correctly, what I'm understanding is that what you're doing differently is taking these large data sets that have a lot of information and trying to make sense of it. Are you using machine learning and AI in that approach?
Absolutely. So nowadays, I mean, AI is a huge umbrella for what it is. I like to say that we use whatever tools are necessary to get the job done. And that spans math, physics, biology, data science, AI. We essentially take the data that we have and we hit it with every imaginable model that we can think of, a lot of which we make ourselves. And what we find is that AI is absolutely great at doing some types of analyses that are very standard and straightforward. And it essentially lets us explore the space of possibilities much faster. So every step that we used to take, which was just a single step, is now actually functionally acting as 10 steps, just because these tools are in the back of our fingertips, underlying all the efforts that we're doing.
That's really interesting. I mean, it's such a powerful tool. Now, are you using that tool for basic research as well as translational research? And what areas are you moving into as far using what you're learning to develop vaccines or identify particular patient populations for maybe new vaccines or any vaccines that are already on the market?
Great question. So the name of the game nowadays for my lab is all about personalized medicine. And everything that we do is translational in the sense of we try to take these huge vaccine studies that people have been doing for decades And now we're trying to ask, of the 7 different types of flu vaccines that you can get this year, and the hundreds of potential vaccines that we could make next year, which ones will be the best for the population? And what would happen if, for example, we don't just have one type of flu vaccine next year, but we have multiple different types? How do we split the population? Maybe 20-year-olds and below should get vaccine 1, whereas 21-year-olds and above should get vaccine 2. And how do we make these splits so that we best cover the heterogeneous responses that we see everywhere? Because we know that the current vaccine technology that we use just doesn't work for everyone equally well. So how do we make things as best as possible?
Well, that's great. So in that business model, as it were, are you using patents to get some of this technology out of the lab and away from the bench into the marketplace and to help better treat patients?
Good question. So at the moment, we're at the step before patents where we're trying to deal with the data. We're trying to show the patterns in the data, show what happens whenever you combine all the data sets from the past 20 years. And I believe very strongly that data needs to be open and public. And whenever I get 5 new data sets, I immediately want to spit them out to the world and say, hey, here are 5 new data sets. Go do with it all the amazing things that you all like to do. And every lab will have their own skill set and way of analysis. So on the data side, at the moment, we're just putting it all out there, combining data sets, showing how they talk to one another. But as we're driving more towards these computational machine learning approaches that say, for you individually, what do we think is going to be the best path forward for you? How do we think your immune response will react? That gets into the territory where we are starting to think about, can we patent this and is it useful? And at the end, will it help make this thing distributed much better and much more practically in the world? So we're getting there to the point where it's starting to become a real possibility and a real decision. But at the moment, we're letting the data be open and free and wild.
That's interesting. So you've done so much so far, and yet you're still so young. So at this point in your career, what is your definition of success?
Good question. So I go back to my roots at the end of the day, and my view of a good, successful project is one where you take other research groups and you turn the airship around, if you will. You take people who are doing something and they say, oh, that is so interesting and intriguing. I'm going to use the results or use the insights developed by those results to do something different, better, faster, higher impact. And whenever you have that collaboration, when you see that other people are using your tools and are excited by them and are making a change, that to me is a definition for success, societally speaking. In terms of the science, I want to always be able to predict how experiments will happen before they happen. That's really the end all be all. I don't want to go into data and say, look at what happened. It was very interesting trends that happened here and there. I want to be able to say, here's a small chunk of the data that you all collected. Now let me predict the remaining 90% of it without even needing to see it, because our models are so good that we can now infer what's going to happen in these experiments. So those are kind of my twin goals for everything that we do.
That's amazing. That's really amazing. So what are your plans for your lab in the immediate future? I mean, what is the big question that you see that is really the hardest nut to crack right now in this area?
So good question. What the future for my lab will be in is going to be a split, in that we're highly specialized and will continue to work in the flu field because there's a lot of energy and excitement in flu at the moment. The NIH is pushing very strongly for universal flu vaccines, which means you don't get a shot every year, but rather you get one shot and it lasts for your entire life. And this is a moonshot goal. It's light years beyond what we currently have, and it's going to take radical new thinking and new approaches in order to push us in that direction. So getting more into the space and understanding how personalized vaccines can help in it will be a big thrust that we do. But at the same time, because we're computational, the lessons that we learned from flu can translate not just to other viruses like HIV, SARS-CoV-2, and others, but it can also translate to other spaces. For example, allergies are also mediated by antibodies or anti-venom effects, or things like understanding phage therapies, or just in general, looking at biological studies and predicting how they will respond at the end of the day. So my lab is going to be flu plus X in the next 5 years. And I don't exactly know what X is going to be, but it'll be influenced by the many amazing people around me here at LJI and at UCSD and around the world, as well as just by where we can make the biggest impact and what our interests are.
So this is all very interesting, and thank you so much for your time today. But if we want to learn more about what you're doing and what your lab is doing, where can we find more information?
So LJI.org will have excellent information, not just on my lab, but the other fantastic folks at the Institute. And there's a bunch of information there on the projects that we currently have undergoing, what we've done in the past, what the teams look like, and a vision from our president on where we're going in the future. It's a very cool site to visit.
Well, thank you so much for sharing your exciting research with us, and we look forward to learning more of your future successes in the years to come.
My pleasure. Thank you very much.
Thank you. Thank you for watching Innovate Law. We hope you found our discussions enlightening and inspiring. Join us next time as we continue to explore the latest trends and share compelling stories on the cutting edge of innovation.